Yeah, thanks so much, Jose Carlos, Sabrina, all the members of the organizing committee. It is great to be here and to be sharing this information with colleagues on a subject that is all near and dear to our hearts. Okay, and I also appreciate the fact that Harris sort of set the context of population fluctuations among sea urchins globally. And I'm going to focus on a particular system off the Atlantic coast of Nova Scotia that I've been studying for 30 years now. And this system has undergone a number of fluctuations. It repeatedly flips from between these two, what I'll call alternative states of the system, kelp beds and sea urchin dominated barren grounds. So my goal will be to present a broad overview of both past and ongoing research that's focusing on the role of disease in particular in driving these changes in ecosystem state, and then to examine how climate change is influencing these transitions. Okay. So this thing changes, the slides, right? Okay, now let me start with a brief outline. I'll begin with briefly explaining the theory of alternative stable states, and this, by the way, is a legitimate acronym. It is found in very prestigious journals, and it's a lot simpler to simply abbreviate it as such. So I'll talk about the theory of alternative stable states. I'll present evidence of these states in the Nova Scotian system, because this is a key issue now. What is the evidence that systems exhibit these dynamics? I'll talk about disease as a driver of transitions, and in fact, the whole cyclicity between these states that exists along the Atlantic coast of Nova Scotia. And then I'll talk about a statistical relationship that we've discovered between outbreaks of disease and hurricanes, and finally how climate change is affecting these dynamics. Okay, so the notion that ecological communities can exist in different configurations in the same place at different times dates back to theoretical work back in the late 60s, early 70s. And there's been an accumulating number of studies and an increasing interest in alternative stable states, particularly because of anthropogenic effects that are changing environmental conditions and causing communities to shift. And the coral reef system is a key example of this. Implicit in this notion is that an established state can persist indefinitely and thereby exhibit stability, and it's arguable how we define stability. It's been defined as persisting greater than the generation time of the dominant species in a community. And also there's this notion that perturbation and stochastic factors will drive transitions from one state to the next. And if we look at this graphically, and I think this paper by Scheffer et al. In Nature in 2001 is a particularly good example. They reviewed evidence for alternative stable states globally, and they present this nice graphical depiction of a community, how a community or an ecosystem can respond to changes in external conditions. And so, whoops, sorry. So we see here that there could be a very gradual, almost linear change in the state of the ecosystem. So this would be the equilibria under a variety of changing conditions. Alternatively, you can have a very pronounced change at a critical level of conditions, or a system can exhibit these alternative state dynamics. So what can happen is that once a threshold condition, or what's sometimes called a break point or a tipping point, is reached, then there's a very abrupt transition to an alternative state. And what's important here in terms of the switching between these two states is that to reverse, to go back to the pre-existing state, requires quite a pronounced change in a reversal in the conditions. You don't simply follow this curve back, but there requires a reversal in conditions to another tipping point in which there's an abrupt shift. So in this intermediate range, there are two equilibrium conditions that can exist under these conditions. And so this difference, this forward and backward shifts occurring at very different critical conditions is called hysteresis, and that's what really defines alternative state dynamics, as opposed to a phase shift, which is demonstrated here. And it's also important to know that severe perturbations can drive a system from one state to an alternative state in advance of even reaching this tipping point. And I'll argue that disease in the system that we work in is one such perturbation. I also find it informative to look at stability in terms of these stability landscapes, these ball and cup diagrams. So the community or the ecosystem can be considered as this ball that sits in a basin. And the deepness of this basin is a measure of the resilience of that community. So when we superimpose these stability landscapes on this state response curve here, we can see at the two extremes of the conditions, we have different states existing only in a single equilibrium, a single basin of attraction. And in these intermediate conditions here, there is a potential for two different states with each having local stability to coexist. And these humps then are the thresholds that need to be crossed so that a system can go from one state to the other. And here we have a perturbation driving a system from one state to the other. So that's the theory. And basically, there's been a lot of empirical evidence that has been gathered for ecosystems throughout the world, for both freshwater, deserts, open ocean. Coral reefs are very trendy now in terms of alternative state dynamics. And interestingly, among the drivers of these state shifts are things like grazer kills, often due to disease. Climate change is often evoked as a driver of transitions between states. Even hurricanes here. So I'm going to argue that the kelp-bed ecosystem, certainly the one off the coast of Nova Scotia, and other systems globally are also good examples of systems that are exhibiting alternative state dynamics, and the challenge is to try to identify the drivers of those dynamics. So to set the kelp beds in a global context, this is a general map that's certainly incomplete now, but this shows in the green bands here the distributions of kelp beds. These are found in cold temperate waters globally. They are very abundant in upwelling areas. This is because kelps require lots of nutrients. And they generally are occurring in relatively shallow waters, down to about 25 meters, sort of on a rough average. And this is due to, in part, substrate limitation that require hard bottom to attach, and also due to light limitations. So here's Nova Scotia in the Northwest Atlantic here. And interestingly, this is the Gulf Stream, so we have this very strong boundary current that influences our local oceanography, and that'll play in later in the story. Okay, now what I'll begin with is a pictorial overview of the kelp beds off of Nova Scotia, so that you can get some idea of the nature of this ecosystem and the players in it. They are very much kelp beds. They're not like the magnificent kelp forests on the west coast. These are prostrate canopies. There's no floats that suspend the fronds above the bottom. Basically, this bed is in the order of a meter or two high. The fronds might be 2 meters long. And they're not diverse. These are the two main species, Laminaria digitata and Saccharina latissima, that keeps changing its name. It's probably changed its name 3 times since I've been studying this system in 3 decades. And in any event, these are extremely rich, luxuriant habitats, high rates of primary productivity. These are structurally complex, biogenic habitats, and as a consequence, they harbor numerous fish and invertebrates. This is the green sea urchin, Strongylocentrotus strobaciensis, and this is a grazing front of Strongylocentrotus. These are absolutely spectacular features. When you come across them, you just can't believe them. So this is just probably about a 2-meter area here. The densities of Strobaciensis in these fronts average between 200 and 400 per meter squared. They can be up to 600 per meter squared. That means that the urchins are just piling up on top of one another. And if you broke apart this pile, you'd find bits of kelp. They feed cooperatively. They climb up the kelp stipes and onto the blades. They weigh down these blades, and then they consume the blades. And they can then consume everything, all the understory algae, invertebrates such as mussels and so on. Everything is consumed, and what are created in the wake of these advancing fronts is barren ground. This is rock encrusted with coralline algae. And these fronts can advance at remarkable rates of up to 3 to 4 meters per month at times. And these are all shots taken in the study areas that we've been monitoring for decades off the coast of Nova Scotia. So when this transition occurs from kelp beds to barrens, there's a tremendous loss of primary productivity by 3 orders of magnitude. And of course, there's a loss of all sorts of kelp-associated fish and invertebrates. So these are very dramatic transitions in community state. And what's left are these barren grounds. And as Harris mentioned, the urchins don't die after they've consumed all the seaweed. They subsist, grazing the coralline crusts and the cover, the microalgal cover, the microbial cover, including the propagules of kelps and other large seaweeds that they basically nip in the bud. They consume these before they have a chance to grow. And there's always a fringe of kelp left in the very wave-washed shallows that the urchins simply can't get at because they have to climb up the fronds, and they can't do that under very strong wave forces. And so there's detritus that's being produced, drift algae that's coming off of those fringe populations. And so you can have these vast barrens that might extend hundreds of meters, even a kilometer off the coast, where the bottom very gradually slopes. And then in other areas where there's very steeply sloping, you have a rather limited distance until it gets down to the sandy bottoms that these rocky areas typically grade into. But throughout, there are these extensive populations of sea urchins and very few algae, only a few very resistant forms. Seaweeds, that is, fleshy algae, persist in these barrens. Now, occasionally, or it used to be occasionally, there were mass mortalities of urchins that have been documented since the 1980s. There's evidence that they might have occurred prior to that, but they've been scientifically documented since 1980, actually, and that's the record that I'll be talking about today. These are conspicuous events. Huge numbers of urchins die. It's in the same order as the diadema mortality. About 98% of the urchins die in the shallow subtidal zone. And they accumulate in piles on the bottom. They wash up on the beaches and so on. So these are very dramatic events. And shortly after they disappear, there is a succession that begins with diatoms and filamentous algae, these opportunistic species that grow very rapidly. And this succession culminates in kelp beds within about 2 to 3 years. So the kelp are the competitive dominants. And so you get these kelp beds reestablishing in about that time frame. So the thing about working in Nova Scotia is that there is a fairly lengthy historical record of what has been going on, certainly in the Halifax region and more recently along the entire coast. This really began with pioneering studies by the late Ken Mann in the late 1960s. And so we have a good record of the fluctuations between these community states. Back when Ken began his work, they were observing urchin populations destructively grazing kelps, certainly in the area around Halifax, and then that was extended to the rest of the coast. So we had destructive grazing going on in the late 60s, early 70s, the formation of the barrens that persisted until 1980 when the first outbreaks of disease began, and there was a series of recurrent outbreaks, as we'll see, and this led to the succession, algal succession that culminated in kelp beds, and then the whole thing repeated itself back in the 90s. And we've seen about 3 or 4 of these shifts between states in the past 30 years. So they occur at approximately decadal timescales. And this slide summarizes these transitions and the various processes that are at play. It's pretty detailed, so I'm just going to draw your attention to a few important concepts here. Firstly, that these transitions, these changes in state are happening in the shallow subtidal zone, down to a depth of about 20 to 25 meters. And this is a depth that is defined by the seasonal thermocline. As water warms in the summer and fall, it becomes stratified. And below this depth, there are constantly cool conditions, and the importance of this will become clear momentarily. So on this axis, we have depth that defines what we'll call the transitional zone, where these state shifts are taking place, as I've just described, and the deep zone, where there are persistent barrens. And what I will argue, the factor the sole factor that drives these transitions in states are outbreaks of disease that cause mass mortality that allow kelp beds to regrow. Following that, there's a whole series of processes that are quite predictable, quite deterministic, that will result in urchins forming aggregations, urchins that persist in this deep area. This is a thermal refuge for urchins from the disease. It doesn't afflict them in these deep waters, but they'll eventually, as the kelp regrows in this transitional zone, urchins form fronts at the deep margin of these emergent kelp beds, and that's when we see an urchin outbreak, and there are all these feedback mechanisms that will accelerate the outbreak and also then impart stability to the kelp bed state. We can depict these changes. So really, it's the waxing and waning of urchin populations that are driving these phase shifts, and we can depict changes in state from barrens from kelp to barrens and back to kelp and so on in relation to urchin density. And so if we say begin in the kelp bed state, this occurs when urchin densities are very low. And as urchin densities increase beyond a threshold for destructive grazing, which is about 2 kilograms per meter squared. These are empirically derived thresholds based on observation. Then what happens is that the urchins consume, they reach these high densities, they feed cooperatively, they pull down the kelps, they advance in these grazing fronts, they consume the kelp bed, and this results in the transition towards barrens, which can persist indefinitely, but it doesn't. There are outbreaks of disease that cause a precipitous loss of urchins, a reduction in urchins, and if this reduction is severe enough to reduce their densities by an order of magnitude relative to that grazing threshold down to about 0.2 kilograms per meter squared, then you get algal succession, the reestablishment of the kelp bed, and so on. A subsequent, this kelp bed will succumb to destructive grazing at high populations of urchins, and then shift to the barrens, at which point somewhere along the line another disease outbreak will happen. And as these disease outbreaks become more frequent, the transition to the barrens basically never occurs. The kelp bed, there might be an increase in urchins, fronts might be migrating up from the deep and driving this transition, and then that will be stopped by an outbreak of disease, and so the kelp bed then rapidly reestablishes. And when urchin host, this is the host population for this disease agent, when that's very low, that disease agent might be introduced and have no consequence. And so what I'm going to argue is that kelp beds are becoming stabilized through increasing incidence of disease off this coast. This is another way of looking at alternative state dynamics where we're relating ecosystem state against urchin density. So at low urchin densities, you have a kelp-bed state. Go beyond that threshold for destructive grazing, and we have a shift to the barrens. And then if urchin density decreases, for example, by disease, past this other threshold, then we have the shift back. This difference between the threshold is this concept of hysteresis that I talked about. And Scott might show this diagram for the system that he's been working with, and he actually plugged in data that showed numbers of urchins and so on quite effectively. All right, so now I'm going to talk about the nature of this disease that has such catastrophic effects on urchins and by extension results in changes in ecosystem state. And as I mentioned, these are really conspicuous events. The urchins are totally incapacitated by this, this pathogenic agent. It destroys their muscle tissues. The first signs is that they lose attachment to the rocks. Their peristomes, their mouth openings gape. They accumulate on the bottom in large numbers, and then they begin to lose their spines and rot. So these are very conspicuous events. And as a consequence, we're able to map the distribution of these events. Both through our own scientific surveys, but also because an urchin fishery was established in the late 80s, early 90s, and this was managed in a manner that the fishers had to keep track of their populations, and so we had good records of where urchins were dying due to disease. So this is a map of the spatial pattern of urchin mortality during the big years of mortality. This is the range of mortalities, and the gray bars are areas of partial mortality, where enough urchins were left that the system didn't flip to the kelp-bed system. It didn't go beyond that lower threshold. And the black bars are areas where basically 98% of the urchins were killed off, and it flipped to the kelp-bed. And there are gaps where in previous years urchins had been eliminated. So really, in the early 80s, we had this interesting recurrence of outbreaks of disease and mass mortality. And then there was a hiatus, and then in the mid-90s, we started seeing outbreaks of disease again. And then in 1999, that was the big one. That extended along the entire Atlantic coast. This distance is over 700 kilometers, killing off urchins everywhere. And this was the end of the fishery. It is still conducted in a few areas, including around Halifax and down here around Shelburne in these small pockets. But most of the rest of the coast, we believe, has gone over to kelp beds, and there are very few urchins. In terms of spatial patterns, there's also that depth limit that I talked about that is a thermally mediated depth limit where urchins are not affected by disease below 20 to 25 meters. Now, if we look at these temporal patterns in relation of abundance of urchins in relation to temperature here, at two sites where I began working in the early 1990s, and we continue to work at some of these sites to date. So the top panels show changes in the density in relation to the temperature cycle, and what you see, what was very prominent, is that these disease outbreaks invariably happened around the peak in the temperature cycle. What we also saw was that the severity of these outbreaks varied from year to year in relation to the peak of temperature, the temperature maxima, and the duration of warm temperatures, which was even more important. So at these two sites, this is sort of at an exposed headland at the mouth of this large bay here, and this is a more protected site within this bay, within St. Margaret's Bay, that I'll be talking more about later on. We see in '93 there was localized mortality much greater at this exposed site than at this more protected site, and it was partial mortality. You can see urchins surviving these outbreaks. There was no outbreak in 1994, so it isn't that this happens every year. And then in 1995, there was a big outbreak that caused widespread mortality at both of these sites and along much of the Atlantic coast of Nova Scotia. So clearly, temperature was playing, from our observations in nature, temperature was playing a major role in mediating these disease outbreaks and determining both the temporal and spatial patterns. So we began conducting experiments very early on in the piece, in 1983, to examine the role of temperature in transmitting this disease by a waterborne route. And so what we did, quite simply, is we took urchins and we put them, we had to go pretty far afield. We had to go into the Bay of Fundy to collect urchins in areas that we knew there was no evidence of disease, and healthy urchins, that is. And so we placed them in aquaria, and we supplied these aquaria with water that was flowing over infected urchins that were clearly moribund and exhibiting the symptoms of this disease, which we hadn't even identified as yet. In fact, we weren't even sure that it was disease as opposed to temperature per se that was affecting the urchins. So we had controls where we put healthy urchins in these tubes. The water flowed through these tubes and into these tanks. And what we found, and then of course we ran these experiments at different temperatures, and what we found was that at temperatures between 16 and 20 degrees, which characterized the annual peak in the temperature cycle off Nova Scotia, what we had was very rapid mortality and all of the animals dying. So within about 1 or 2 weeks, 50%, none of them, all of them were dead within that period of time. And the rate at which they would exhibit the symptoms of disease and then die was very temperature dependent. So here we see the time to 50% morbidity. We use this as an index of the transmission of this disease. It's very high. The time at these higher temperatures, it takes a very little time for the urchins to get sick and die, and then as the temperature decreases, this time to morbidity increases exponentially. At these high temperatures between greater than 12 degrees and up to about 20 degrees, all of the urchins die in these waterborne transmission experiments. But we found at 12, there was some survival. About 20% of the animals survived, and it took a long time. It took about a month for only half of the urchins to exhibit symptoms. At 10 degrees, the urchins were totally asymptomatic. They were exposed to infected urchins, but they didn't develop symptoms of the disease. And we could take animals that we infected, say at 16 degrees, and place them in 8-degree water, and they would recover. So if we got them before they died, but were exhibiting symptoms, they would recover at 8 degrees. At 22, we make urchin bouillabaisse. They controls, everything dies. That's the thermal limit for Strongylus introtus. So this reinforced that temperature was really important in controlling the disease, and it explained everything about the patterns that we were observing in nature. It explained the seasonality. So here's temperature at varying depths from 5 to 36 meters in 1999. Remember, this was the big year, and we observed mass mortalities, and we looked at temperatures, and it was an unusually warm year. And so, again, the mortality occurs in the seasonal peak. It's very dependent on the duration of that seasonal peak. That is, the severity of the die-off is dependent on the duration. As temperatures cool towards the fall into late October, November, they descend below this pathogenic threshold of around 10 degrees, and the populations, any surviving urchins at that point will recover. And of course, it also explains that depth limitation to the disease, because below about 25 meters, the temperatures are below that pathogenic threshold. So it explained a lot. And, but we still didn't know what the pathogenic agent was, and so we really concentrated our efforts into understanding that. And I had a postdoc in my lab, Gwyneth Jones, that found this amoeba in the tissues, of moribund urchins, and found it was a species of paramoeba that didn't seem to conform with the existing species, and so she considered this a new species. And a big breakthrough at that time was that we were able to develop monoxenic culture techniques and azenic culture techniques to isolate this amoeba and to culture this amoeba, and we were able to prove Koch's postulates, which was a major breakthrough. We could inject urchins basically cause disease and then isolate the amoeba from those diseased urchins and reinfect other urchins. And most recently, with the molecular tools available now, I've collaborated with a geneticist, and I never thought I'd show a slide like this in any one of my talks. I really don't understand any of it, but apparently, but for this audience in particular, because I know similar work is going on here, we found that Paramoeba invadens was in fact a true species, and it was most closely related to Neoparamoeba branchiphylla, which is a paramoeba that's been isolated from species of urchins here. And we're going to argue that we should drop, this guy's going to argue that we should drop Neoparamoeba and go back to Paramoeba as a genus. This is Alistair Simpson that I'm referring to. So I'd be interested in, this is a paper that we're just revising for diseases of aquatic organisms. All right, so getting back to the etiology of this disease, we've established this nice temperature regulation, that this is a disease process that is the dynamics of which are very tightly regulated by temperature. And we were able to grow these amoebae in culture, so then it was logical to look at the effects of temperature on amoebic growth rate. So these are the two graphs that I'd shown you before. And I've added here how growth rate's measured is not all that important, but we can see this is the growth rate of amoebae in culture, and the maximum growth rate coincides with these peak temperatures off the coast of Nova Scotia when we see these disease events occur. And we also thought that maybe this species was coming in from warmer waters through the Gulf Stream and so on. So we had this very high temperature here, and the populations just crashed. So somewhere beyond 20 degrees, and there's an upper thermal threshold that suggests that this is not a tropical amoeba by any means. But more interestingly for us in Nova Scotia and for work that we're currently doing is that there's this lower thermal limit, about 5 degrees, where the populations are not sustained, there's no growth. And below, this was at 2 degrees, I think. This is work done with my first PhD student, Joanne Gellett, who was a microbiologist. So I was an ecologist, and I was accumulating all these friends that were, parasitologists and microbiologists and so on, because this was the big question. What was causing the disease outbreaks? And at 2 degrees, these populations just crash, so there seems to be a lower thermal limit. Why is that important? Well, because this suggests that this amoeba is a non-indigenous or an exotic species. Certainly if they can't survive at 2 degrees, there's no way they're going to overwinter off the coast of Nova Scotia, at least what the coast of Nova Scotia used to be like, where temperatures routinely drop below 0 in winter and can be -1, even lower than -1 in some cases. It's bloody cold. So if the amoeba couldn't overwinter, then it would have to be reintroduced every time there was a disease outbreak. And indeed, we could only find, the only place we've recovered these amoebae is from the tissues of moribund urchins during disease outbreaks. In the same area where urchins might have survived, when you sample in the next year, but there's no evidence of disease, we can't recover paramoeba. So it suggested that these things would be introduced, die off over winter, and have to be introduced again. All right, so what was bringing them in? And so I thought about this some time ago, and I looked at different oceanographic mechanisms that might create sufficient advection to bring an amoeba in from distant areas if it couldn't live along the coast of Nova Scotia. And one of the first things that I thought about were hurricanes, which are huge mixers. And you can see this shot. This is Hurricane 1. This caused a lot of devastation when it hit Halifax. It came right in and smacked into Halifax in 2003. It caused huge destruction and even death. And so here's Nova Scotia, and for scale, this is about 700 kilometers along the mainland coast, and this is one, a massive system. And of course, this creates a lot of horizontal advection due to the wind forces of the hurricane, but also vertical advection where the deep water is being moved up. And Nova Scotia is a spectacular place to watch these passing hurricanes as they cross across the North Pacific. This is another hurricane that occurred much later in the year, and this is an intertidal area where we've done a lot of work, but normally this is all exposed rock. This is beyond the intertidal, and you can see huge amounts of water pouring in through the coast, but these explosions of waves that are like 100 feet high or more. And that happens to be me. And this was a student that was way up in the safe position on the rocks where everybody else was. And they said, look at that idiot down there. I'll get a picture. And then when he saw that it was me when I came up, you know. Anyway, so I'm serving a scale there at any rate. Spectacular things. Okay, so this brings us to the killer storm hypothesis. So I had this notion, and I published it. It's just sort of a wild idea, along with some other wild ideas about oceanographic and meteorologic events that could link these outbreaks of disease along the coast to, advection processes. And hurricanes were just one of these things. And we had assembled some information by that time that looked like these disease events were preceded by strong hurricanes in the years that they occurred. And then Juan hit Nova Scotia. And we had been doing an experiment off the coast. And after Juan had passed by, our urchins began dying due to disease. And I thought, well, that's interesting, but still it's sort of an N of 1 after I proposed this. And then a few years later, again, we terminated another large experiment because urchins were dying due to disease after another Hurricane Bill. And so at that point, I got together with my former PhD student, who's a math head and modeler, Jean-Sébastien Lozanguet, and we decided that we should look at this in a more rigorous statistical fashion, which we did. The interesting thing about these disease events that followed the hurricane, the timing was exactly that, almost exactly to a day or two, of what we would predict based on the temperature dependence of this disease. So if you said the disease was introduced at the point at which the hurricane was closest to the coast, and you knew what the post-hurricane temperature was, you could predict when you should be seeing 50% of the urchins exhibiting signs of disease, and it was spot on. So there was something there. And we had this fantastic record of mortality events over this 30-year period from 1980 to 2009, again, because there was a lot of scientific activity along this coast and fishing, urchin fishing. So we were able to get information from the fishers, especially post-1999. And of course, you can get excellent hurricane track information from the National Hurricane Center and so on. So that's a fantastic record. And the Canadian government, the Department of Fisheries and Oceans, monitors temperatures at different depths all along this coast. So we were able to compile all of this information. And we developed basically a statistical model. We used logistic regression that related whether a disease event happened or not in relation to characteristics of these storms, their intensity, measured as wind speed, their proximity of the coast, things that would affect the degree of mixing felt along this Atlantic coast of Nova Scotia, and of course the post-storm temperature. And what we were able to do is identify a series of candidate storms in each of the 12 years when we had these mass mortalities. And we found that the timing of the mass mortality in relation to when the storm passed by the coast really matched that predicted by the post-storm temperature, as I've just mentioned. And so what these basically are plots of our model that relates this probability of a mass mortality to these storm features. This is distance from the coast on this axis. This is wind speed, so you can see the very warm colors indicate that there's a very high probability when you have an intense storm that's close to the coast, and very low beyond that. And these black dots are our candidate storms that really provided the best fit. This series of storms provided the best fit to our regression model. And we also looked at it in terms of temperature as a dummy variable. it would get a 0 or 1 depending on whether it was below a thermal threshold or above the thermal threshold, and we let the model determine what that threshold was, and came up with 12 degrees, which was very satisfying. And so you can see there's only one storm here that had a threat that occurred below the thermal threshold, but it was close to the coast and was fairly intense. The gray dots are simply storms that occurred after our candidate storms, and so we don't know to what degree they might have contributed to the mortality event. The disease had happened. We were just interested in the first storm that elicited the disease. Okay, and we also then did trend analysis using a quantile regression over this 30-year period, and what we found was that there was an increased frequency of storms with the highest probability of mortality. That is, the highest quantiles were showing a positive trend. And that means that the most deadly storms have become more deadly, and it's a trend that is continuing. And associated with this trend analysis, we found there was an increased intensity in terms of wind speed and proximity to the coast of these strongest storms. And there was an increase in post-storm water temperature. This is a graph. This is the most simply interpreted of these graphs. This is just average water temperature during the hurricane season, between August and through to November, over this 30 years. And you can see this is a significant increase in water temperature. And this rate of 0.7 centimeters per year is higher than the average rate of warming in the North Pacific. So climate change is real. It's happening. And for urchins, it's dangerous. So we've seen this increase in the intensity of the strongest storms that is linked to ocean warming. The people that really study hurricanes have shown this, and in fact, they've shown that the frequency of these strong storms is expected to double in this century. So our prediction then would be there's increased frequency of killer storms this increased frequency will limit the resilience of sea urchin populations in the shallow subtidal zone and actually maintain the system in a kelp-bed state. So to test this, we've been conducting this large-scale transplantation experiment now, which will carry on 5 minutes? Okay, this will be the short ending, which will carry on over 5 years. And what we're doing is we're transplanting urchins from a site where we haven't seen much evidence of disease over the past decade into St. Margaret's Bay, where we have seen Hurricane Juan and other hurricanes, 2 events in the last decade. And we're placing the urchins in these cages here that we're hurricane-proofing by wedging them in amongst boulders. And these are various sites, between 5 and 7 sites within St. Margaret's Bay. At one of these sites, we place them in deeper water where it's colder. We have a back transplantation in our source population, and we monitor morbidity and mortality. And our predictions are that the morbidity and mortality that we see at these various sites due to this disease should occur after storms of a high probability of mortality based on the storm features and the post-storm temperatures. And we also would predict that in the deeper water, the rate of mortality would be much less. Okay, this is a very complex diagram. I'm going to just sort of ask you to trust me, because to go through all of this, we're monitoring significant wave height, temperature, and the proportion of moribund or dead urchins at our various sites. So in 2010, 2011, the short message is conformed entirely with our hypothesis. These are the probability of mass mortalities. It's not significantly different from what we established as the mean of the 12 storms based on our long-term record. They occurred around the same periods of time, and what we see at exactly the predicted time, or within 2 to 3 days, we're starting to get mortality, or we get 50% mortality on average across our populations, much less in the deeper water. This mortality here was simply due to handling a different way that we transplanted the urchins. So really it was at this point that we saw these increases. There's the storm, there's the mortality. It's happening as we expect. We support the model. Then, in August of 2007, a beach walker in the Halifax area, sent me this photograph. And this is a mass mortality. This is what they look like. These are urchins washed up on the beaches around Halifax. No hurricane. And I'd pretty much written most of this chapter by then, figuring that this was, I knew what the conclusion was. So then there was a lot of, lateral thinking that had to go on. And I guess I don't have enough time to really get into the details of it. But there was a wave event around the 1st of August that preceded these observations on the 12th of August. This was not related to a hurricane, but could have caused some mixing. There was also a period of really warm temperatures. So we had more than 50% morbidity at Halifax at this point in time, and we ran to our sites at the mouth of St. Margaret's Bay, and in fact, they were starting to die as well. But there was some partial mortality here and no mortality. And as time went on, we did finally get a hurricane, Leslie, which actually was significantly, the probability of mortality was significantly lower than that for our candidate storms. But it probably just served to mix things even further once a pathogen was in the neighborhood. And so then we got this secondary effect at the right time at our source population. So now our source population was compromised. The urchins died at this place that we had transplanted the animals from. So we had to think of other ways. And I went back to that 1997 paper and I also proposed that things like warm core rings that spin off the Gulf Stream that can entrain huge amounts of water. The rings don't come onto the shelf, but they do push water across the shelf. And this might have been a mechanism that explained the warm waters that preceded this outbreak of disease and could act to transport the pathogen. And so my grad student, Colette Fian, who has helped me in this along with Jean-Sébastien, her boyfriend was in oceanography. He had just done an internship in NASA. So you make these connections. And he got us this data. This is like, I don't know, I think this is confidential. It can't go out of this room or something. I don't know. It's highly classified. Anyway, this is the most recent stuff of sea surface temperatures in this area off of Nova Scotia, and we see this band of warm water approaching the coast in late July, early August. We picked this up on our temperature loggers. It was around this time that we had this mass mortality. Could this be a water mass that was bringing in the pathogen? There was a second wave of even warmer temperatures. These are in the order of 20 to 22 degrees, ridiculously high temperatures for Nova Scotia. And this was when our control site went. So there is a suggestion. The other thing that happens, we also have triggerfish off Nova Scotia now. I was diving with triggerfish just in August and trying to catch them. They were sometimes they wash up dead, but these were very lively and active. The year before, we were diving with mola mola. We're getting flying fish, seahorses, all kinds of things. So if these organisms can be advected to the coast of Nova Scotia at this period of time, a small planktonic amoeba probably could be as well. That's a possibility. Wow, I'll just briefly mention that JS is a master at predictive modeling. He does all these simulations. And so what we did is we hindcast from our data record of 30 years based on storm characteristics, and we were able to calculate what we'll call the cumulative probability. The probability of having a mass mortality of urchins in any given year based on storm characteristics and water temperatures. And what we found was that prior to 1980, the first time we scientifically documented mass mortalities, there was a very low probability of these happening due to storm events, less than 0.3. Then we had this rapid rise over the period of record, and then our forecast up to about 0.9. So that's a 90% chance if you're going to have a killer storm in a year, and then beyond that, it basically is the end of days for urchins. Just very high. And we can break that down into the different components. The distance from the coast, you can see that over this period of time. The storms are getting closer to the coast. The size of the bubbles is the wind speed, the intensity of the storm. The color of the bubbles is whether the post-storm temperatures are above or below the critical threshold. So, so really, this is a projection of how climate change is really going to result in a loss of urchin populations, because there will be a disease event every year. A loss of urchins in the shallow transitional area. I don't have time for this, I'll just gloss over it, but what we're also looking for is what's happening at the trough of the temperature cycle, and the possibility that temperatures are now exceeding the lower tolerance limit of the amoeba, and the possibility that the amoeba may be surviving off the coast. So I showed you this graph about amoebic growth rates. And so here's the information about that peak period, and you can see that as time is progressing, this is this 12-degree thermal threshold. At 12 degrees, urchins will die if enough time elapses at temperatures above 12 degrees once they're infected with paramoebiasis. And you can see that there's a progressive increase. So that's what's happening at the peak. And the double whammy is that at the trough of the temperature cycle, our winter temperatures are warming. And so this is what we believe to be this 2 degrees thermal threshold. The amoeba dies below 2 degrees. And what we're seeing is that on average over this period of time from 1980 to present, we have an increase in winter temperatures. We're repeating the experiments, and we're finding that urchins that appear to be dying in cultures at these low temperatures won't recover at 0 degrees, but around 2 or 4 degrees, they'll eventually recover if temperatures warm. So this would be my short ending, and I have a number of conclusions, but perhaps I'll let you reach your own conclusions. I do want to just skip through and just the acknowledgements here. I'm sorry if I've gone over time. That's sort of the cast of thousands that have helped. That's another story. That's the end of this story. But hang on, let me just get to that last slide of acknowledgements. This is Colette, our killer. She's such an innocent lass, but her business is just constantly killing urchins in Nova Scotia with the disease. And this is Jean-Sébastien Lozonguet, and guess who? So special thanks to my co-authors on this venture. You know who this is. Yeah. Okay. Thank you very much.